Good afternoon, everyone. I would like to thank first the organizing committee for inviting me as a speaker to this conference. I am Amnah Alsayyar, an early stage researcher at Dasman Diabetes Institute, and today I'm going to talk to you about our research on how TNF-α increases the production of IP10 and its role in breast cancer progression. Breast cancer incidence affects 1 in 8 women in their lifetime at any age following puberty, and the prevalence rates tend to be higher later in life. The WHO by the end of 2020 estimated that around 7.8 million women were diagnosed with breast cancer in the past 5 years, making it the most prevalent type of cancer worldwide. If we look at the incidence rates in Kuwait, we can see that breast cancer covers around 20.6% of the total number of cancer cases in 2020, making it the second highest type of cancer here in Kuwait. Incidence rates, or the cancer incidence rates, mainly are linked to several risk factors like gender. For example, women tend to be at a higher risk than men in breast cancer. Genetic and family history are linked because if you have a family history or a close relative who had breast cancer, your chances will be higher. Age, as I mentioned earlier, the prevalence rates tend to be higher later in life, and obesity and breast density. So, with obesity, for example, it can be linked to cancer because obesity is associated with several metabolic disorders that can cause um, some dysregulation in cellular function and performance, which in some specific cases can lead to cancer. So, how immune cells act in the tumor microenvironment? So, the tumor microenvironment consists of a diverse population of cells that includes, like we see in the figure, of course, the cancer cells and the different types of immune cells. According to research published, these tumor associated immune cells can possess either tumor antagonizing or tumor promoting functions. So, what happens basically is that the tumor antagonizing immune cells within the tumor microenvironment will target and kill the cancer cells in the early stages of tumorigenesis. However, the cancer cells seem to eventually escape from the immune surveillance and inhibit the cytotoxic function of the immune cells. If we talk specifically about the breast cancer microenvironment, now the breast cancer cells are secretory by nature, and their secretory activity can be modulated by a variety of inflammatory stimuli that in turn induces the production of pro-inflammatory cytokines and chemokines, which can promote tumor activation and cancer progression. So, what is the role or what happens to chemokines in a tumor microenvironment? So, chemokines are soluble, small molecular weight proteins that direct or guide inter and intra-organ movement of immune cells. So, they basically facilitate the movement of cell and their migration as well. They are important for many biological events such as embryogenesis, wound healing, tumorigenesis, and metastasis. They can be classified into four groups based on the location of the first two cysteine residues in their protein sequence. The infiltration of immune cells in the tumor microenvironment is a key factor in cancer prognosis, and chemokines play an essential role in guiding the migration of both activating and suppressive immune cell types. Now, the migration of immune cells into tumor tissue is unpredictable because the solid tumors, for example, like breast cancer, are heterogeneous and do not have a defined anatomy, making it more difficult for making it for difficult also for the chemokines to actually know what they're doing, and they will assist in actually um, cancer cell migration. That's why in a tumor microenvironment, they will actually promote tumor formation, and they will play a major role in metastasis of the tumor cells as well. Okay, so this figure here just shows the types of immune cells and each chemokine linked to it. And the most chemokines, according to the papers published, that are involved in many tumors are CXCR4 to CXCL12 and CCR7 to CCL21. Now, our chemokine of choice was CXCL10 or IP10. We wanted to know what its role in breast cancer. So IP10 is a pro-inflammatory chemokine that was first identified and reported in interferon gamma-treated monocytic cells. IP10 is involved in the regulation of immune responses through the recruitment and activation of leukocytes and endothelial cells, as well as the different types of immune cells like natural killer cells, T helper cells, macrophages, and beta cells as well. It is highly expressed in various inflammatory diseases like type 2 diabetes, cardiovascular diseases, multiple sclerosis, neurodegenerative diseases, and as well as cancer. It is also known to be a pro-tumorigenic chemokine that has been strongly correlated with the progression of breast cancer, given that it is more largely secreted by malignant tissue than non-malignant tissues. Next, we tried to link it with TNF-alpha, as the title of the research states. So TNF-alpha is a pro-inflammatory cytokine that is secreted by macrophages and play a key role in glucose metabolism. The disturbance of TNF-alpha pathway can lead to several metabolic disorders like diabetes and in some cases cancer. According to research, this specific cytokine displays characteristics as both tumor promoter and tumor suppressor, as in it can affect tumor vascularization and initiating apoptotic cell death, but can also promote cell proliferation. When produced at high levels, it supports cancer development, especially in patients with solid tumors like breast cancer. So, how all of this is linked? So, as I mentioned, both TNF-alpha and IP10 encourage tumor progression if produced at higher levels. The role of TNF-alpha in IP10 production in breast cancer is yet not clear. And so we hypothesized that, and we wanted to check, could actually TNF-alpha induces IP10 production in MCF7 breast cancer cells? And if so, which signaling pathway or pathways are involved in it? So first of all, we wanted to check and confirm and see if TNF-alpha actually induces the production of IP10. So first we treated the cells with TNF-alpha and we left them incubated for 24 hours and then done PCR analysis on them. And as we can see from figures A and B, that the mRNA expression levels and the protein expression levels as well were significantly higher compared to the control when treated with TNF-alpha. Okay, that's the IP10 expression, meaning that TNF-alpha actually induces IP10 production. Then we wanted to check if IP10 production is dose dependent. So we treated the MCF7 cells with different doses of TNF-alpha, as we can see here in figure C, and we observed that it actually is dose dependent. So the higher the doses of TNF-alpha, the more IP10 expression we get. And we also confirmed the IP10 expression and intensity through confocal microscopy, and we can see here that when we treat it with TNF-alpha, we can see more IP10 expression than the control, which is like in the green color, almost double. Okay. After we confirmed that actually TNF-alpha induces the production of IP10, we wanted to check which pathway or pathways it is actually involved in. So from our studies, we know that P38, JNK, and ERK-MAPK pathways are known to be regulated in different cell types through the exposure of pro-inflammatory cytokine TNF-alpha. So we basically treated the MCF7 cells with different inhibitors specified for these specific pathways. Okay, so and as we can see here, when we treated with the MK, ERK, and P38 MK, ERK, and P38 pathway, we didn't see any much difference in terms of the IP10 production compared to TNF-alpha here when we treated. But one pathway was significantly downregulated, as in from the IP10 production, which is the JNK. Like we can see here at the mRNA and at the protein level. And we also did confocal ANA just to confirm and see, and we can see significant reduction in the IP10 expression when we inhibited actually the JNK pathway compared to the others. So that basically showed that the IP10 production through TNF-alpha depends on the JNK pathway, and the other pathways didn't show any significant results. Next, we wanted to further confirm if it actually depends on the JNK pathway, the JNK pathway. So we transfected the cells with JNK siRNA, which, as we can see in figure A, resulted in a significant reduction in the JNK mRNA expression levels compared to the control. And with the IP10, we can see here a significant reduction in mRNA and protein levels when we inhibited the JNK pathway. So this basically confirms that it depends. The IP10 production, the TNF-alpha, depends on the JNK pathway. Furthermore, because c-Jun is located downstream of the JNK pathway, and according to our hypothesis, we thought, okay, if we inhibited c-Jun, we expect a reduction in actually the TNF-alpha induced IP10 production. So we transfected the cells with c-Jun siRNA, which of course resulted, as we expected, a significant reduction in the c-Jun mRNA level compared to the control. And this basically confirms also the same thing here for the IP10 production at the mRNA and at the protein level. And this basically shows and confirms that the reduction, that the induction of the IP10 by TNF-alpha requires c-Jun kinase signaling. Last but not least, and as a final confirmation of our results, we decided to determine the phosphorylation and activation of JNK in TNF-alpha. And as we can see, we did Western blotting, and we can see here that the JNK phosphorylation and c-Jun phosphorylation when treated with TNF-alpha was significantly higher. Okay, but a significant decrease was observed when we inhibited the JNK pathway, which is basically aligned to our previous results. In addition, and furthermore, we also did a SAP-K and JNK kinase assay was performed to using glyzates prepared from the TNF-alpha treated and vehicle treated MCF7 cells. And our results, like we can see here, showed that the kinase activity of JNK and TNF-alpha stimulated MCF7 lysates was significantly upregulated compared to that control lysates. So this was basically confirmed our previous results. So in conclusion, and what we concluded from our results is that TNF-alpha stimulation induces the IP10 transcripts or protein expression in MCF7 breast cancer cells through the mechanism involving the JNK or c-Jun mediated signaling, which implies that targeting the JNK c-Jun kinase of MAPK pathway may have a significant elevating TNF-alpha IP10 mediated breast cancer progression and metastasis. Our future direction from that we would take, and we would like to know that if this co-stimulation with TNF-alpha and interferon gamma have a synergetic effect on IP10 induction in breast cancer and other cell types. This figure here just summarizes the pathway that we did and our results. So we can see here TNF-alpha goes through the phosphorylation of JNK and c-Jun pathway, which eventually leads to production of IP10.